So if you look at the studies that have been done with chemotherapy, suddenly the numbers are basically 1/10 of the numbers for pleural mesothelioma. No randomized study, all phase 2. And you can see that the response rate at best is around 25% for all studies, whatever agent you use. This was a study we conducted at MD Anderson when I was still over there. I want to mention the Columbia approach, which was promoted by Dr. Robert Taub at Columbia in New York. And this is a very complicated multidisciplinary treatment of selected patients with peritoneal mesothelioma. It starts with a debulking surgery, not a total peritonectomy, followed by intraperitoneal chemotherapy, sequentially with 4 doses of doxorubicin, followed by 4 doses of cisplatin, followed by huge doses of interferon gamma. And I'm not even sure this is commercialized still. Then once the treatment is completed, a second surgery to debulk what's left over, followed by a heated perfusion with mitomycin and cisplatin, followed by whole abdominal radiation treatment at 3,000 centigrade. The overall survival in this selected population is 70 months, which is really excellent, with about 70% that are free of disease at 3 years. In this group of about 30 patients, 6 are still alive without disease at 86 months. So to be aggressive is a good thing in selected patients that are going to be able to tolerate the treatment. Going next after chemotherapy, what else can we do? Well, the 21st century is the era of biological therapy. So let's see what has been done for mesothelioma with biological therapy and immunotherapy. Today, there is really no proven efficacy of any biological therapy in mesothelioma, but there is hope. And then there has been some older study of immunotherapy, but this is best in patients that have a minimal bulk of disease. So these are studies of interleukin-2 or interferon in a very small cohort of patients, showing at best a 20-month overall median survival with response rate that is very variable. There was a very interesting study from Stephen Abelda in Philadelphia doing gene transfer of an interferon beta genes. There was a phase 1 study. The maximum tolerated dose was this number of viral particles. The treatment induced a transient lymphopenia, which you would expect with interferon. They documented 7 patients where the transfer was successful, and they had immune response elicited in 7 of these patients with different stimulation of the immune system, sometimes T cell, NK cell, humoral responses, and the clinical benefit rate, which was mostly stable disease, was 40% in that study. The biological target of peritoneal mesothelioma are listed here. The 2 most important ones is EGFR and mesothelin. So EGFR has been overexpressed in 60 to 90% of patients, with about 30% of them having an EGFR mutation. However, when we tried studies of TKI, such as Tarceva, in patients with mesothelioma, there was 0 response rate. That was a study that was done in SWOG. So we don't really know how to interpret this data, and there is still actively studying the expression of EGFR and how to circumvent any problem with treatment with selective inhibitors. Mesothelin is very interesting, and I'm going to show you here what really mesothelin is. It's basically the product of a mesothelin precursor protein that gets cleaved in a megakaryocyte potentiation factor, and that's probably why patients with mesothelioma have a thrombocytopenia in the mesothelin. And so the mesothelin is the receptor for CA125, which it binds in a very specific manner, and we know exactly which amino acid are binding to the mesothelin from the CA125. So there are now strategies to try to inhibit this binding side of CA125 to mesothelin using monoclonal antibodies. And both of them are also shed in the blood. So mesothelin is overexpressed in about 70% of patients with mesothelioma. It's also overexpressed in ovarian cancer and pancreatic cancer. And patients that have overexpression on the tumor, 80% of them will shed some mesothelin in the serum. And so there is a monoclonal antibody against the CA125 binding domain called SS1 that are currently being used in trials. And I'm going to show you two phase 1 trials. The first one is the antibody linked to a toxin for Pseudomonas aeruginosa called SS1P. 24 patients were treated. The maximum tolerated dose was 25 micrograms per kilogram per day, continuous infusion for 10 days. Constant level of SS1P were obtained in the plasma, was one partial response and some minor response. This drug is now going in a phase 2 study for pancreatic cancer, unfortunately not mesothelioma. The second study is using MORAP-009, which is an antibody directed against mesothelin. Again, 24 subjects, 13 with mesothelioma. There were very minor adverse events. Maximum tolerated dose is 200 milligrams per square meter. Best response is stable disease in 6 patients. And again, this is going to go for pancreatic cancer phase 2. So what are the treatments that are available to patients who have failed first-line or are recurring after first-line treatment? Well, most of it is extremely experimental. What we know is that patients that responded to first-line treatment will do better with a second line than patients that did not respond in first line. Whether we should use a doublet versus a single agent is not clear. There is really no cure at this point, and I always recommend a clinical trial. Now, this is a challenge for peritoneal mesothelioma. No pharmaceutical company wants to open trial for peritoneal mesothelioma. There is not enough business in this disease, unfortunately. So we do rely on foundation and rare tumor orphan drug coverage. Phase 1 studies is still a very good approach because we need new treatment for this disease, and targeted therapies are also things that we should study. So in conclusion, still many questions remain about how to take care of peritoneal mesothelioma. What is the role of surgical debulking? It's still not clear. We know that we can probably cure some patients, but we don't know how to select them. We need to study that further. How to better use the drug, intravenously, intraperitoneally, which are the optimal drugs? The list is relatively long. What is the biology of mesothelioma, and how can we target crucial biological mechanism in something that we will need to study much further in the future? And then immunotherapy, many progresses have been done in other diseases such as melanoma. I think these discoveries should also be applied to mesothelioma. And then I'm going to make a plea that when a study is open for mesothelioma, it's usually an exclusion criteria to have a peritoneal mesothelioma. And I'm going to make a plea because the disease is basically the same. All patients should be able to enter those studies. Thank you very much.
So you say they're the same. I've heard some people say they have a different etiology. Is there a different etiology?
No. Unless you have one of those papillary, well-differentiated or multicystic mesothelioma, if it's an invasive cancer, it is the same as pleural mesothelioma. It behaves a bit differently because of the anatomy.
And there really isn't any markers, you're saying, that are...
Well, we're looking at mesothelin and, as the main marker for mesothelioma.
You said earlier, at the very beginning of your talk, there's no threshold for safety. I mean, how do you quantify them? How do you lack of quantify them?
It's difficult because a lot of people don't even know they were exposed. No, when they die, but it's too late, you can measure them. No, there are some labs that are going to measure the amount of asbestos in your body, but no, what good is it once you're dead? Yeah, so I mean, it's more a legal problem than a medical problem at this point. Yeah. Okay, well, thank you.